Thanks, Fanny. And I don't see exactly how many people I have joined so far, but happy to see that so many people took the time of your day to join this webinar. So my name is Niko Markkinen. I work as a food contact material testing expert here at Measure Labs, and here to talk to you today about NIAS testing, how it's done, how the testing is done, how the results are evaluated, and what is the regulation behind it all. So short about myself. My background is actually in food sciences. I'm a Doctor of Science in Technology in Food Development, defended back in 2021 from University of Turku, 200 kilometers west of Helsinki. And soon after that, joined Measure Labs as a testing expert, first both about food and food contact material testing, but quite soon afterwards only have been, let's say, 100% focusing on food contact materials. And what I do on a day-to-day basis is I help our clients to make sure that the food contact materials and articles they make or import comply with the relevant regulations, be it EU, FDA, some Asian markets as well. And for those present who are not familiar with our company, we are a testing service provider located here in Vallila in Helsinki, Finland. And the basic idea behind the company is that we consolidate key testing services from over 900 laboratories from all over the world, which enables us to provide testing services for a whole range of different sectors. Food and food contact materials is only one of them. We also support clients from semiconductor industry, municipalities in terms of water or environmental testing, and a host of different range of testing services. And when it comes to food contact materials, it's not only testing that we provide, that we also provide different sort of support in documentation preparation and in designing testing, assessing different matters that they might face. So I would like to say that it's an all-encompassing service for food contact material manufacturers or importers. And also want to mention that the headquarters is in Helsinki, Finland, but we also have a site in Cambridge in the UK and also one in Palo Alto in the United States. About the agenda of the day, start from the basic stuff, what NIAS mean in practice, what is the regulatory framework behind it, especially in terms of plastics, how NIAS are tested. Once you have the results, how those results are then evaluated, ending with services that we have available here at Measure Labs, and also then addressing your questions, either ones that are brought up during the presentation or those that were sent to us during the sign-up. So what are NIAS? I imagine that the acronym is familiar to people, but it comes from non-intentionally added substances. And in very broad terms, it means anything present in food contact material or FCM that has not been added there purposefully. And NIAS can be divided in different ways into different categories. And one of them would be its origin, being side products, breakdown products, and contaminants. Side products typically could mean something that it's a side product from the polymerization process, that it generates something that it's not supposed to. Second would be breakdown products, that certain monomers or additives break down either during the manufacturing process or later on during the use and the storage of the material. And the lastly would be contaminant, either already present in the raw material itself or an environmental contamination during the manufacturing chain. Other way to categorize NIAS would be depending if it's known or if it's already identified or whether it has been assessed. And let's say the optimal category is that the NIAS has been identified and assessed. For example, it would be that we know that in recycled PET there could be benzene present, and then we test for the presence of benzene, so then it's identified and it's assessed. Then the second category would be identified but not assessed. So we are able to predict that this material could have certain type of NIAS, but we haven't done any assessment for it. Third category would be detected but not identified, which essentially would mean an unknown substance in the NIAS profile. We see that something is there, but we don't know what it is. And the worst case category would be not detected. So we just don't know it's there. We do what we are supposed to in terms of the targeted and non-targeted analysis, but we just don't capture it for one reason or another. But it's there. We just don't know it's there. And in terms of analytical approach, the NIAS testing can be divided into predicted testing of predicted substances, which means that we analyze for that substance in particular. And the other one would be to test for unpredicted substances, because we don't know what we're looking for. We have to use non-targeted screening. And these two terms will be elaborated later on in the webinar presentation. So important question is, why is testing in NIAS relevant in the first place? So I have divided this very simply in two, let's say, realities: regulatory reality and migration reality. The migration reality would be that the Article 3 from the framework regulation says that the food contact materials and articles shouldn't endanger the health of the end user. And when it comes to plastics, the Article 19 specifies that there should be a risk assessment done for NIAS. Then again, on the migration end, or what is present in the FCM itself, it is estimated that most food contact materials contain certain level of NIAS, and most of the substances that migrate just in pure amount of substances are unknown or unpredictable in a way. And when we put this in the larger scale, we see that the potentially migrating substances vastly over exceed the amount of listed or authorized substances, or those that are specifically regulated in EU. So put all of these together, it means that the NIAS analysis and risk assessment thereof is a crucial part in evaluating safety of food contact materials. So what then are typical NIAS? It's an important question as well. So I mentioned these three categories already in the previous slide, but in a recent review article from 2021, in the big picture, it was concluded that the main NIAS typically present are breakdown products of additives and monomers, especially those of antioxidants. This would be followed by side reaction side products, and lastly being oligomers. This is of course very situational, so this is just in the grand scale of things. And especially when we talk about using recycled materials, either 100% recycled or mixed with virgin materials, contaminants can become a relevant source of NIAS. So here are a few examples of NIAS that are generated from these antioxidants. And why is this the case that these are such a common source? There are two factors played here. Basically, the substances are used so commonly because you have to protect the plastic or the rubber or any type of organic material from deterioration caused by oxidation, UV, etc. And so basically these substances take the hit instead of the other plastic materials. But what this in turn leads into is that the materials very commonly have these degradation products present as NIAS. So here are just a few examples that, let's say, IRCAFOS 168 can oxidize into, or the phos, it doesn't matter, can generate oxidized IRCAFOS 168, but more as it degrades into further, then can generate this Arvin-4 substance that is then very commonly detected in various food contact materials. Here are a few common examples of NIAS that are detected depending on the material. Primary aromatic amines can essentially derive from the typical sources being azo-dyes and polyurethanes. In case of polyurethanes, have to be an aromatic polyurethane, so aliphatic polyurethanes don't generate these. So if there are free monomers in polyurethane-based coating or adhesive, it can react with water to generate these primary aromatic amines, or azo-dyes could then degrade to separate these substances. Then we also know that when PET is being reprocessed to make recycled PET, this process can then generate acetaldehyde, benzene, or 2-methyl-1,3-dioxolane. But there's a whole range, so these are just examples. Then one class would be additives. Unfortunately, the risk assessments or assessing safety of oligomers is currently lacking. The German BFR has done risk assessment for polyamide or polyamide 6 and polyamide 6,6 in particular, those cyclic oligomers, and we will discuss that also a bit later. The free or the degradation of the material of free monomers present generate these larger structures. But this applies to also a whole range of different polyesters in particular. Once the toxicological assessment has been done for these oligomers, the cutoff value is considered being 1 kilodalton, because it assumes that anything larger than that is not absorbed by the human gut. An exception would be PFAS, where the cutoff value is being considered as 1,500 kilodalton. In terms of contaminants, here are a few examples. Let's say in recycled plastics, you could potentially could have phthalates. If you have PVC contamination, you could have bisphenol, either a carryover already in a recycled material that enters again into the recycling process, or you have some materials there that have bisphenols present, such as can coatings based on epoxy or adhesives based on epoxy. And this also highlights the importance of NIAS analysis for recycled materials, because then you have the kind of NIAS load already in the material kind of entering reprocessing, where you generate then more NIAS as well. So it's not only the contaminants present, then the degradation of those intentionally added additives also builds up or could build up in the material. So there was the examples of different NIAS that can come from all sort of different sources. So then what would be the regulatory framework once we consider NIAS? In the big picture, Regulation 1935/2004 applies to all full contact materials. And what is especially relevant for myself, at least, is the Article 3, that in a sense says that food contact materials and articles should not release materials or substances at a level that endangers human health, brings about unacceptable change in the composition of food, or has impact on the appearance, odor, or flavor of food. So this kind of sets the backbone, let's say, that we have to know what NIAS are present and that they don't migrate at a level that could endanger human health. But this doesn't really give you any kind of pipeline to go through to do that assessment. So then for plastics especially, we have the Regulation 10/2011, which I will be referring as plastic regulation here on out. So basically that's built up in a way two parts. You have the union list that in Article 5 it says that you are only allowed to use substances that are authorized under a union list, and some of them then have the migration limit assigned to it. But since, as highlighted before, NIAS can be a whole range of different substances, most of them would be unpredictable. So then there is an exemption that NIAS don't need to be included in Annex 1. So having NIAS present that is not listed in the union list doesn't make the material automatically non-compliant. However, what was recently added under Article 15 is that there should be more transparency when it comes to NIAS. And the wording specifically is that if there is a risk that the NIAS is present at a level that could cause non-compliance in the final article, this has to be disclosed to the next in line in the manufacturing chain. And what has been around for a long time is the Article 9, which in a sense says that there has to be a risk assessment done for NIAS. So it has to be specific here. It doesn't mean that you have to test for NIAS. It doesn't mean that. It means that you have to do risk assessment for it, and testing is one part of that. Yeah, but it doesn't kind of mandate the testing, let's say. What was also added recently is the requirement for a high degree of purity, and I have noticed that this causes some confusion. That, and I have to be precise here, that the high degree of purity doesn't apply to the plastic itself. It applies to the substances that you use in the manufacturing of food contact materials or plastics intended as food contact materials. And the whole background there is to ensure that the materials used there don't carry, let's say, a NIAS load on its own that then carries over into the final material. So you have to do a risk assessment of that. So there are 4 options for it. Unfortunately, the case here is a bit kind of in motion at the moment, that it's not the current available methods for assessing or doing migration tests of NIAS from solid articles are not directly applicable to powdery chemicals or liquid solutions or anything like that. So some development has to be done on there, how you would actually assess the NIAS effectively from the substances themselves. So that was the regulatory framework in a nutshell. Then next we move on to discuss how on practice you would test NIAS. So here I have collected a few possible options. Doesn't mean that these are the only options. There are a whole range of different ways of doing it, but let's say that these are the most common ones. And this slide I want to highlight is that there are several factors that you have to take into account, and especially that you cannot use one specific method to cover everything, let's say. And one of the key factors would be the volatility of the substance. Is it extremely volatile, like something like solvent residues, or is it non-volatile, a very large molecule that you cannot use GCMS to analyze, or is it an inorganic impurity, heavy metals, etc. So then you have to combine all sort of different techniques to do the final assessment. The GCMS is the most, let's say, common, most robust way of doing it for a whole range of reasons. And ICP-MS testing is typically done in any case, because the testing of restricted elements is part of essentially every food contact regulation. The headspace analysis or the emission analysis is in a way situational, depending if you have material that is based on solvents, that either it's a printing ink based on solvents, or if it's a coating applied with a solvent, then that is expected to evaporate, and then you just test to make sure that there is no solvent residues left as a contaminant in the material. That's a bit like a borderline case if that's NIAS or not, but that's here or there. What I also wanted to highlight with these methods, whether they are targeted or non-targeted, and that kind of goes back to the initial slide, that the headspace analysis is typically targeted, meaning that it's specifically done for a certain solvent or a scope of solvents that we look. Emission analysis can then be both, that then you can also look into outside of the typical scope of the headspace analysis. But what are the kind of driving horse in a way of this NIAS analysis would be then the GC and LCMS non-targeted screenings, which are then, because from the non-targeted nature, and maybe I should specify what that means in practice, is that once you analyze your simulant with the instrument, you get a range of peaks on a chromatogram. And then you have to answer 2 questions. What are those and how much is there? and then you do a risk assessment thereof. So the first question is that, or the second, let's start with the second question, like how do you know what's there? So you could have a host of common reference standards that you compare to. You could do a library match. So in GCMS, you have the breakdown of the substance, and you have a mass spectrum, and then you compare that to the library and see if you have a match. And if you have a match in a good quality, you can be pretty confident that it's this substance. But it's not always possible. It might turn out that we don't have a good clue what it could be. So then we could look into the scientific literature if there is a match for this particular mass spectrum. And in case that it means it's semi-quantitative, or quantitative means that you quantify the result using the actual substance that you analyze. So let's say that for styrene, you use styrene to quantify how much styrene is coming out from your material. But when it's semi-quantitative, you have one or more internal standards where you have in relation to that substance the quantification. So you have some understanding of the magnitude of migration, but it's less certain than quantitative result would be. And what I wanted to also highlight that the typical reporting limit with these non-targeted methods is 10 parts per billion in terms of migration into food. So these techniques are very effective in doing what they do, but there are definitely certain limitations. The first one being that since the result is semi-quantitative, which I already mentioned, that there is some uncertainty in the results. Another problem would be that because it's a kind of catch-all method, that it's not validated or specified for any substance in particular, there could be some variation in ionization between the substances, which means that the uncertainty in the semi-quantitative results then also varies from substance to substance. And due to this semi-quantitative nature, the measurement uncertainty is not possible to be determined. And then the second problem or challenge would be then the identification of substances, because optimally you would always have a reference substance to back your identification, but since the amount of NIAS possible is so high, there is no way you can have a reference substance for everything, so you have to rely often for the library match. But if the match is poor, then there's uncertainty if you are looking at the right compound or not, most likely not. And then you could then go into literature to find a match. That's very time consuming. If you find an article, it might be behind a paywall, because unless you are working for a research institute, you don't typically have access to a lot of these articles, even though these free articles are getting more and more common. And then the last problem would be that if the substance is completely unidentified and you don't find a match in literature, let's say, then how do you work around with that situation? And one additional limitation with these techniques would be that it's not suitable for everything. Like I discussed earlier, if the contaminants are heavy metals, you cannot see them with CC or LC. But there are also a range of different substances where you cannot really apply these non-targeted approaches. And here is a kind of collection of some of those. So for example, in case of primary aromatic amines, because in the Annex 2 of plastic regulation, the limit for carcinogenic or primary aromatic amines that have been associated as CMR substances, the migration limit is 2 parts per billion. But if you have a method where the detection limit is 10 parts per billion, so that just doesn't work. So if you have a risk of primary aromatic amines in your product, then the only way to work around that is to do using a method that has been validated to accommodate that low detection limit. And in other substances, it just could be that it's not technically very viable method to do this non-targeted analysis, or it's not precise enough for that purpose. So for, let's say, oligomers, additive breakdown products, certain contaminants. These are very effective tools, but not for everything. Okay, so then we accept that this is the best we have at the moment. They are very effective tools, but we just have to accept there are certain limitations, and in some cases we have to do complementary analysis doing targeted methods. So then we move on. How do we design the testing actually? So we have to consider the simulant we want to use, the test conditions we want to use. Universally, 95% ethanol is considered the worst case simulant if the intended use to use with all food types. Of course, if you want to only intend the contact with acidic foods, then you would test with the associated simulant, which would be then 3% acetic acid. So of course, it doesn't make sense to test with 95% ethanol if the intended use is much less harsher than that. But one thing I wanted to bring up here, that not with every material, it does make sense to always use 95% ethanol, because with certain materials, it's a known issue that this simulant causes swelling. So then perhaps considering using isooctane instead would be a good idea. Then again, if the contact temperature is very high, something that is intended in the oven for 200 degrees, with ethanol and isooctane, you can only use maximum 60 degrees. So then there's a discrepancy that if you test 60 degrees for 10 days with ethanol, how does that compare to a tray that is in the oven at 200 degrees for 2 hours? So that's a lot of uncertainty. So then it would maybe make more sense to test with Tenax instead, where you can put the tray actually at the equal conditions for 2 hours at 200 degrees. So it's always a balancing. Or possibly you could do both, but typically that's not what people want to do. So they want to have one option to do it. So in that case, it could be considered that using Tenax would be a more appropriate plan for high temperature applications. So how do you then choose which conditions to use? There are tables present in the plastic regulation already in the Annex 3, but that doesn't kind of describe every possible scenario. So there are some cases where it's not very specific. So then there is the Joint Research Center's technical guideline, in particular for kitchenware articles, which nowadays also includes tables for paper and board materials as well. And those can be helpful references, which I also use very often. Okay, then we have done the testing, we have designed the conditions, we have the results, then what next is what would be discussed. So it would be essentially a tiered approach where you start first to look if the substance is present in the union list. If there is a migration limit, you use that migration limit. If there is no migration limit, then you apply the overall migration limit, which would be 60 milligrams per kilogram of food. It has to be noted that this is not always very apparent, because there might be some groups there that you don't find a match perfectly for the CAS number because it's part of another group there. So that should be mindful about. And also that for some substances, isomer salts or esters can be accepted even though not in the annex itself. So if that option is not available, then you could look further into regulation available in EU. What's a common reference is Annex 10 from Swiss ordinance, which is a national regulation of Switzerland, which has a very comprehensive positive list, and that includes a lot of substances that are not listed in union list. And if that then again doesn't provide satisfactory reference, then you could look further if there are any action limits or guideline limits presented on a national level by BFR from Germany or DTU from Denmark. And then you use that. You could look if there is a limit in drinking water that could be then calculated in the migration limit into food, or if there are some levels set up, what would be the tolerable daily intake, then it could be also calculated on an ad hoc specific migration limit. And if none of those options are available, then you do what is called use so-called TTC approach, where based on the structural properties of the NIAS, it's assigned from Kramer class 1 to Kramer class 3, which have specific guidance limits for maximum daily exposure, which can then again calculate it into migration limits. So here are the threshold limits in the EFSA TTC approach. What I wanted to mention is important here, that in order to use this approach, certain conditions have to be met, so-called in a EFSA TTC decision tree. You have to go through and check different factors. But because of time constraints, I'm not going to go in depth into that. And typically, most of you who work in the manufacturing sector, this work is already done on your behalf by the laboratory service provider. So typically, this is not something we have to concern, but it's good to be, I think, aware of. So these threshold values, being milligrams per kilogram per body weight per day, can then be calculated into these migration limits. And the idea there is that a person weighing 60 kilograms consumes 1 kilogram of food packed into or being in contact with the food contact material that was tested every day, 365 days of the year, basically. And so then, but it's a very robust way of getting some type of guidance limit in cases where no other options are available. Kind of going back into that tier, in case the EU regulation or the Swiss or other national regulation doesn't provide any limits for the risk assessment, I wanted to bring up a few examples of using this different alternative references. And for polyamide oligomers, for polyamide 6 and polyamide 6,6, there is some limit for 5 milligrams per kilogram of food, which is our opinion by BFR, which is a very good reference. So that, because then you don't need to use the Kramer classification, this is much more precise. And in particular for those antioxidant degradation products, for many of the most common ones, these migration limits have been calculated based on drinking water positive list limit, or later on then maximum threshold concentration at tap. So basically you have that value and then you feed that into a certain formula, and then on the other end you get the migration. And then it's a bit different because we mostly talk about plastics, but just as an example, for cyclosiloxanes, which are the most typical NIAS in silicones, there's an action limit by Danish DTU for D3 to D8, then would be 12 milligrams per kilogram in adults, but this meaning migration, obviously, and in children 2 milligrams per kilogram. So before heading to conclusions, there are two more matters I wanted to kind of discuss here that might be something that's causing some headaches, or you would like to get some more insight into. Would be if you see a non-compliance in the results, what would be the next steps? And the second one would be to address the discrepancy between the limit for genotoxic substances, which is 0.15 parts per billion, while the reporting limit for the non-targeted methods is 10. So then there is the discrepancy there. So I wanted to discuss those two matters lastly. So starting with, if you see a non-compliance, what next? So these are just suggestions. These are not kind of like rock solid guideline to do it. This is our, let's say, best technical expertise opinion on this matter. So because it's a very large, complicated topic, there are many ways of doing it. I'm not saying it one way or more correct than that, but maybe just as a food of thought, I wanted to bring this up. So the first one being to determine batch-to-batch variation. Is the non-compliance, is it one-off or is it a consistent issue? Then, since there are in the screening methods, there are uncertainty with the identification and there are uncertainty with the result. So then you would do targeted analysis of the NIAS to confirm the identification and get a more precise value for it. This is a beautiful thought, but the practice is that targeted methods are available only a very small number of substances. So it's a pretty much coin toss if you have methods available in any laboratory for that specific NIAS. Then one point would be to concern if the test protocol overestimate, because you tend to, the migration is always done in certain particular ways where the simulant is in contact with the article or the part of the food content material. But if it's, for example, like a cap that is not typically in most cases, it's not in direct contact. And if you see a small exceedance, then you could maybe make a justification that on most cases it's not in direct contact. So even if we see a small exceedance in this setting on a bigger picture, we don't consider this a non-compliance. Obviously, an important thing would be to identify where that NIAS is coming from. Is it a degradation of an additive? If it's a degradation product of antioxidant, can you do something about it? Can you reduce the dosing? Can you do something in your processing that would reduce the amount of this degradation product being generated? Same with oligomers as well. If it's a contamination, where is this contamination coming from? So identifying the source and removing it. Then one mitigating factor would be a formulation change or functional barrier. So let's say that the problem would be on the recycled end of your material, then you just reduce the amount of recycled content and increase the amount of virgin content. Or if it's a multi-layer structure, then you could put that problematic layer, let's say, behind a functional barrier like aluminum. So then you don't have migration into the food itself. So that could be one way that you could use recycled material in the structure without having to worry about the contamination then migrating into the food itself. And lastly, then would be amending of the use of material. If all else fails, then you need to consider that, okay, maybe for this specific material, we cannot use very long migration times. So we cannot make lunch boxes out of it, but maybe we can make cutlery out of it. So then you have substantially lower migration times. And when you do the testing with that, for that particular use, you don't see any issues with the results. So that could be one option as well. And lastly, also, this is also a topic that is kind of living at the moment, and I don't kind of want to give you a vision that this is like an ironclad solution to it, but this is more about kind of wanting to address it, because I imagine it's in many of your heads, something that you're thinking about, and maybe possible solutions that we could have in the future for addressing this. So basically in the annex, sorry, I forgot which annex was it, but in any case, the one that requires the contents for declaration of compliance, that you have to address substances where genotoxicity has not been ruled out, and you have to do the NIAS declaration as discussed earlier. But what I often face as a question is that, okay, you have a migration limit of 10 parts per billion, but I need to address substances below 0.15 parts per billion. So how can I do this? And unfortunately, there is no analytical approach at the moment, to the best of my understanding, to do this. So addressing this matter is more about doing a risk-based approach which analysis can be part of it. But what I wanted to highlight here is that when you look at the wording in the regulation, is that it's specifically about addressing intentional use when it comes to these substances where genotoxicity has not been ruled out. So the regulation doesn't mandate that you have to address NIAS on this sensitivity. And even if the law required it, it's not just technically possible at the moment. But then that as to another question. Then okay, so what about NIAS, that where chenotoxicity has not been ruled out, that migrate below the 10 parts per billion but over 0.50? What's that about? Well, as I mentioned, and I want to reiterate, there is no catch-all analytical solution, but some suggestion to kind of mitigate the risk is that you do a risk assessment on the formulation and processing conditions and use the best kind of scientific and chemical knowledge to think about what type of NIAS could be generated in our product, or if any of those substances this would apply where chenotoxicity cannot be ruled out. And if such concern is raised, instead of doing conventional migration test, one option would be to do more harsher, for example, solvent extraction of the material. So you have a more concentrated sample, so you can see on a more sensitive level what's present there. The only limitation with such approach is that because you extract so much from the plastic or the whatever FCM we are talking about, there is a lot of background noise, so that on the other hand can raise the detection limit for such approaches. One thing being discussed already for a few years now would be to use bioassays, so you don't have to address any particular substance there. So you do bioassay, for example, Ames test for the migration solution as a whole to see if it reacts positively for chenotoxicity or not. The kind of a conflict there is that as these migrations are typically done with 95% ethanol, and if you do chenotoxicity test for that, it always comes back positive because that ethanol solution is chenotoxic. So then you have to dilute it a lot, and then you lose a lot of sensitivity for this method. But I hope that not just this problem, but this problem in general will be solved in the future by people who are much smarter than I am. So looking forward to that. So wanted to kind of close this as soon as possible. So we have answers the questions. Actually, the last one actually was a question that was not initially part of the presentation, but I wanted to bring it up because I think it's something that at least a few people are considering thinking about at the moment. But to kind of summarize the main points of today's webinar, that NIAS are unavoidable, and they are present in almost all or nearly all food contact materials, which can originate from different sources, be it additive, breakdown products, side products, or contaminants. The risk assessment, especially for plastics, is a legal mandate, and then there is a documentational mandate as well nowadays. There is no, let's say, one right way of doing the risk assessment, but the typical approach is as I described, and it's a multi-layer process where you end up with EFSA TTC approach if other suitable references are not available. There is no single method that can cover all NIAS, and some NIAS would require a specific targeted method, one major example being the primary aromatic amines. And what I want to end this webinar with is that often I see that people hope that you could just send your sample to analysis to kind of solve this whole NIAS thing once and for all. But my message is that the risk assessment cannot or should not rely on testing alone. It's about, in the big picture, about comprehensive process that you understand, you predict and address potential risks in your material. You understand the kind of chemistry to the best as reasonably possible, what's going on there, what could be potentially being generated. And what is also important, that there is enough transparency between the manufacturing chain to what could be happening there. So the next guy in line knows what to consider in their own risk assessment. So yeah, that was the contents of the webinar. I wanted to talk to you about so quickly about the services we have available. All of the analysis mentioned today, I mentioned all of them are available from us. These non-targeted screening methods also. And in case there are some strange results or unidentified substances, we can support you as best as we can to also identify those, and that would fall under this extended risk assessment of NIAS. So if you are interested in these services, please don't hesitate to reach out. And in addition to this, we obviously provide the general compliance assessment testing based on EU or FDA especially. Right. Any questions, Fanny, in the audience?
Yes, we have a few questions here that we could go through. Starting from the first one, so there's a question about fatty food applications, and in which the SML tests need to be done using both 95% ethanol and isooctane. But for NIAS testing, can we then choose just one of these simulants, or do we need to test with both?
Typically, I would see that in almost all cases, the test is done with one simulant alone. I would only consider that if there is higher due diligence placed on the manufacturer for some reason, that they have to have more elaborate information available, that you would do with two simulants. But let's say on a practical experience, testing with one simulant is more than sufficient.
Yes. Then we have a question from Marty about rPET, and what do you think of the need of grinding rPET in sample preparation?
Depends what you want to do. If you want to do migration test, I would avoid against grinding it, because then it really depends what you want to do. If you want to analyze, let's say, content of certain type of contaminants, then grinding makes sense, because then you expose more surface areas of the sample, and then the extraction is much more effective. But if you want to simulate migration, the optimal way would be to make a test piece out of that rPET, like a plaque or a bottle or something like that. Or then a second option would be to test from granulates themselves directly. But if you grind it down, then it becomes more like an extraction than migration.
Yes. And then we have a question about VOCs or VOCs and SVOCs, and which limit of quantification is possible to reach in your opinion with non-targeted screening for non-targeted VOC and SVOC?
That one I have to check that one. I don't remember by heart, because typically the targeted analysis are done for the VOCs. This is something that I would like to discuss more detail in my colleagues. That would this be something that would be valuable for our clients in the food contact material testing sector, but it's done less, this emission testing for food contact materials. These are mandated more, let's say, for paints and floorings and things like that, so you don't have emission of formaldehyde or things like that. But it would be very interesting to see. The only kind of limitation there is that in that method, it would be let me reiterate. So the most common cases where I see these emission tests being used are in case you have some kind of defect in any of the batches. You might have a batch that has strange malodor or something like that, and you want to identify the source of the malodor or what substance is it caused by, and then you can identify the source of the odor. So in that case, once you do emission test for a batch that is not affected, and you do an emission test for a batch where you have the defect, and you compare the profiles, and you see that there is some VOC present in the defect batch, and then you check if those VOC have some aroma properties, then you can say, okay, this is coming from this substance, and then that might help you to identify why you have this off-odor present in your material.
Yes. Then we have a question about the Swiss ordinance from Sylvia, who works for a converter. And she asks for the new Swiss ordinance where list B substances is now deleted, and the new GI ordinance to determine the potential migration of 10 parts per billion. Is it possible to use the NIAS approach?
That's a very good question. It really depends on, I would say that if we are talking about printing inks, and if we are talking about intentionally using some substances that are not within the positive list, then to be safe is to adhere to the requirements of the Swiss ordinance. But if it's an NIAS coming from a printed article, then I would use the Kramer classification instead of the Swiss ordinance 10 parts per, sorry, the 10 parts per billion limit. Yeah.
Yes. Then we have a question about the small exceedance of migration limits, and this is maybe a specific question and one that is a little bit case by case, but Sirkku is asking what we consider a small exceedance limit.
Yeah, that's very situational. What is considered small? I would say roughly maybe less than 10%, but that's very hard to say because maybe put that into a bigger picture that in the recent update for the plastic regulation, there now is the formula which you apply to results of specific migration to kind of have this general measurement uncertainty, where you basically don't think that if the average is just a bit over, that it's a non-exceedance, but you apply this generalized measurement uncertainty there. It's not 100% clear for me yet if that is applicable for NIAS results or not, but I will try to learn it as soon as I can.
Yes. Then continuing on the same topic about what to do if the migration limits exceed, how can we determine whether the migration test protocol overestimates the migration compared to the actual use conditions?
That's a very situational, and that usually is something that should be done in discussion with the laboratory, or if you have a product stewardship unit in your company, then with them. So it's very hard to put like very broad specification there because it's very situational.
Yes, that's what I thought as well. Then we have a question from a PVC film producer. He is obliged to test NIAS, but as far as he understands, there is no specified method standardized. So what method would you recommend, and what testing frequency should be used?
Yeah, that's a very good point that wasn't brought up into the discussion, that the harmonization and standardization in NIAS testing is lacking a lot. So every laboratory has, well, the techniques are the same and the basic principles behind are the same, but there is no standardization. So in very general terms, I would always advise any plastic material or article manufacturer that the NIAS testing with GCMS is the most relevant one, and depending on the situation, it's good to complement with the LCMS testing as well, especially in materials where a risk of oligomers is elevated. And the frequency, that's very open-ended questions that varies a lot of different things. I can say that, let's say, by experience, the typical frequency I see is between 2 to 3 years. But to my understanding, there is no kind of maximum limit that you have to adhere to, or minimum frequency that you have to adhere to. I would say 4 years is kind of pushing it, because even though you kind of keep the manufacturing process and the raw materials the same, you kind of small things can change over time. Small things can happen in the machinery, some small things can happen in the raw material that can impact the NIAS profile over time. Then you would kind of see that, okay, after 4 years, maybe it's reasonable to repeat the test.
Yes. Then I think we can take one last short question, or there's actually two people have asked a little bit similar things about the 95% ethanol and why that is considered the worst case simulant and the one that is most commonly used, and does it cover simulant A and simulant B?
So I don't have exact answer why 95% ethanol would be superior to isooctane, but when we talk about NIASes, they most often tend to be more on the lipophilic end than on the hydrophilic end. Kind of going back that the most common ones are these antioxidant degradation compounds, and those are much more effectively migrating into 95% ethanol, let's say, compared to 10% ethanol. There are some, let's say, kind of outliers or exceptions to this, which I would say that by experience, polyamide oligomers tend to migrate better into acetic simulant compared to 95% ethanol. But universally, the 95% ethanol is the best catch-all simulant to do it, because it's a very harsh, more like a solvent than a food. And since the majority of these NIASes are more on the lipophilic end.
Thank you. Then I think we'll start to wrap up. If you can put the last slide with the contact information.
Sure.
Visible.
Yes.
Yes, this one. So it is now 1:00, and I want to say a thank you from my side to Niko for the great presentation, and to all the participants for good questions. And if you have some testing needs, of course, or if you want to discuss further, more specifically about how to test your products, then you can find Niko's contact information here and be in touch that way, or through our website as well, of course.
Yeah, thanks. Completely forgot. Thank everybody for your attention.
So then I think we can end the meeting and wish everybody a nice continued afternoon or day, depending on what time zone you are in. Thank you.
All right. Thank you. Bye-bye.
Bye.